Hello friends, today we are going to discuss the Juxta Glomerular apparatus or the JG complex. Now, if you look in this particular diagram, you will understand that there are four parts or four cells in this particular Juxta Glomerular apparatus. The first cell type is called as the Macula densa. This one. The second one is the JG cells, that is Juxta Glomerular cells. The third one is Extramesangial cells, mesangial cells, or which are also called as Polkissen cells or Lacis cells, or these are also called as the polar cushion. And the last cell type which is present is called the intramesangial cells. Are you getting? There are four cell types present in the Juxta Glomerular apparatus. The first one is the Macula densa. Second one is the JG or Juxta Glomerular cells. Third one is the Extramesangial cells, and the last one is intramesangial cells. If you look at the Macula densa cells, these Macula densa cells are tall, columnar, and narrow cells. These are tall, narrow, and the columnar cells. These are present in the distal convoluted tubule, present in DCT. These Macula densa cells, they consist of centrally placed nucleus. Next cell type is the JG cells, which are also called as the Juxta Glomerular cells. These JG cells, they are nothing but the modified smooth muscles of the tunica media of afferent arteriole. These are the modified smooth muscle cells of the tunica media of the afferent arteriole. These JG cells, they consist of round nucleus. Another most important characteristic feature that you have to remember, that the basal lamina is absent. Basal lamina is absent between the JG cells and the Macula densa cells. As a result, these two cell types, that is the Macula densa and the JG cells, they are intimately connected with each other. The next cell type is the extramesangial cells. These are the cells which are present in between the distal convoluted tubule, afferent arteriole, efferent arteriole, and the vascular pole of the Bowman's capsule. The next cell type is the intramesangial cells, which are present in between the glomerulus. Next point, what is the function of these particular Juxta Glomerular apparatus? Now, if you look at the Juxta Glomerular apparatus, these Juxta Glomerular apparatus, whatever the Macula densa cells present in the JG cells, they perform two functions. They monitor the filtrate volume and they maintain the sodium concentration. Macular densa cells, they perform two important functions. They are responsible for monitoring the filtrate volume and the sodium concentration. When the concentration of sodium drops, it will result into two functions. These macular densa cells, they perform two important functions. They stimulate the JG cells to produce renin. This particular renin is an enzyme which converts angiotensin-I, which is a 485 amino acid long with 33 signal peptide molecule. Now, this particular angiotensinogen is cleaved by this renin from the N terminal to produce angiotensin-I, which is a decapeptide, means it has got 10 amino acids from the N terminal of this particular angiotensinogen, and it is mild vasoconstrictor. Now, the second enzyme, which is called as the angiotensin converting enzyme, which is mostly present in the capillaries of the lung, they convert this angiotensin-I, which is a decapeptide, into angiotensin-II, which is a potent vasoconstrictor and it is an octapeptide. Are you getting? The first macular densa cells, they are responsible for monitoring the filtrate volume and they regulate the concentration of the sodium ions. When there is a drop in the concentration of sodium, two events occur. The first is it stimulates the JG cells to produce the renin. Now, this particular renin is an enzyme which converts the angiotensinogen, which is a 485 amino acid long protein having 33 amino acid signal peptide. Now, this angiotensinogen is converted to angiotensin-I by a renin, which is a decapeptide and acts as a mild vasoconstrictor. AC, that is angiotensin converting enzyme, from capillaries of the lungs, they convert the angiotensin-I into the angiotensin-II, which is a potent vasoconstrictor and it has got 8 amino acid molecules. Next point. Now, this particular once angiotensin II, which is a potent vasoconstrictor, is released, it performs following functions. This angiotensin II, it performs 4 important functions. It causes synthesis and release of aldosterone. Because of this particular aldosterone, there is a reabsorption of sodium and chloride from DCT, that is distal convoluted tubule. Second, it causes the release of ADH, that is antidiuretic hormone, from the neurohypophysis, and this ADH is responsible for reabsorption of water from DCT. And the last point, this particular angiotensin-II, it causes the release of prostaglandins. These prostaglandins, they act to vasodilate afferent arteriole, and this vasodilation of afferent arteriole results in increase in glomerular filtration rate. Angiotensin-II also inhibits the renin, resulting in negative. This is what is the first function of the macula densa, and the second important function of the macula densa is that it increases the blood pressure. Okay, we will revise it once again. We will revise it once again. The Juxta Glomerular apparatus, or the JG complex, it consists of 4 types of the cells. The first one are the macula densa cells, which are present in the distal convoluted tubule. Second one are the JG cells, which are present in the afferent arteriole. Third one is the extramesangial cells, or Polkissen cells, or the Lacis cells, and the last one are the intramesangial cells. Whatever the macula densa cells are there, they are tall, columnar, narrow cells with a centrally placed nucleus. Second one, these are JG cells, are the modified smooth muscle cells present in the tunica media of the afferent arteriole. Extramesangial cells, they fill the space between the distal convoluted tubule, afferent arteriole, efferent arteriole, and the vascular pool, while the intramesangial cells, they are present in the glomerulus. Now, whatever the macula densa cells are there, they are responsible for 2 important functions. First one is that they are responsible for regulation of the blood pressure, and the second one, they are responsible for release of renin from the JG cells. Once this renin is released, this renin converts the angiotensinogen into angiotensin first, and angiotensin first, which is a decapeptide, is converted into angiotensin second, which is a octapeptide by means of an enzyme that is called angiotensin converting enzyme. Once this angiotensin second is formed, which is a potent vasoconstrictor, it causes the release of 3 important substances, that is aldosterone, ADH, and the prostaglandins. The aldosterone is responsible for reabsorption of sodium and chloride from the distal convoluted tubules. ADH is responsible for reabsorption of water, while the prostaglandins, they are responsible for vasodilation of the afferent arterioles. Next point is, whatever the angiotensin is there, it causes the release of negative inhibition of the